Hello, and welcome to Breast Cancer Conversations, a podcast brought to you by survivingbreastcancer.org. I'm Laura Carfang, breast cancer survivor and founder of survivingbreastcancer.org, a nonprofit organization providing community, education, and resources to empower those diagnosed with breast cancer and their caregivers from day one and beyond. Hello, my friends. Welcome to another episode of Breast Cancer Conversations. I'm so excited for today's conversation. I am joined with JP. JP is the founder of Astrin Biosciences, a St. Paul-based cancer intelligence company transforming detection and treatment via AI. Motivated by his father's cancer diagnosis and passion for research, he left his role as Deputy Chief Science Officer at United Health Group to launch Astrin in 2021. JP serves on the board at the Hennepin Healthcare Research Institute and holds a PhD in brain-machine interfaces from the University of Minnesota. Welcome to the conversation. Thank you for taking the time out of your busy day to be on Breast Cancer Conversations. For our listeners, we are joined with JP. I will let you give your formal introductions, but you are the founder and CEO of Astran Biosciences, and today we're going to be talking about a really important topic that I know is, top of mind for a lot of us women, not just those who've been diagnosed with breast cancer, but are really advocating for women's health in general in terms of understanding your breast density and getting screened with mammograms, which we know is kind of like the first line for just kind of screenings and preventative care for cancer in general. But I was diagnosed in my 30s, well before my first mammogram. And, there was no family history. I was a very healthy young adult. And it definitely blew my mind to find out, wow, I have cancer. And since then, there's been a lot of work, over the last, I would say, like 5 or 6 years to really get to this point where people are now being informed of their breast density. It was astonishing to find out a couple years ago that clinics were not required to inform women of their dense tissue and what that actually means in terms of their risk for developing cancer later on, and also for understanding how well actually the mammogram tool is. It is a great tool to the extent of your density, right? And so we're going to dive into a lot of these topics, and JP is going to share with us some really novel work that him and his team are doing with regards to being able to detect breast cancer early, especially for us women that are healthy in our 40s at time of screening, had a negative mammogram, which is great, but there's still risk factors for breast cancer. And so we want to understand what is those risk factors and can we catch them early? Without further ado, JP, thank you so much for being here today. And I'll let you introduce yourself, where you're calling in from, and the extensive background that you have in this space.
Thank you. And Laura, thank you for the opportunity to share some of the work that the team is doing. We stand on the shoulders of giants who not only furthered the science that we are employing to build the kinds of tests that we are working on, but also the giants whose voices made a difference in trying to get the notification law passed last year. So whether it was Nancy Capella or Leslie Yeager or Joanne Pushkin, all of these folks who have been so active in this space to get the law to pass such that women across the country can get their notification and know what density their mammogram reveals.
Absolutely.
A lot of what we do is it starts with trying to understand how cancer progresses. And that's the fundamental insight that when we heard in having conversations with some really sharp folks at Mayo and at Sloan Kettering and other places, this notion that cancer doesn't progress local, then go regional, and then metastasize, but that cancer cells leave the breast early in the process was something that we were intrigued by. And our whole purpose was, could we find these early cells? Could we then reveal the signatures of these cells that will allow us to find their vulnerabilities? These cells, as they're traveling through the circulatory system and the conversation, the constant crosstalk they have with the immune system, could we tap into those conversations and understand the molecular sort of fingerprint of these cells? So these are the types of questions that prompted us to start what we're doing. It also, of course, had an unfortunate beginning. My family, like a lot of other families, is not immune to cancer. We've had several family members who've been diagnosed with cancer, and unfortunately some that passed away battling the disease. And for me, that was a deeply personal mission. So how do we, based on this insight, try and build the best technologies, the best platforms we could imagine to try and answer this question? because it's a shift from how we think about cancer, and could we then now pursue the best science in the spaces. Again, a lot of what we do is built on some fantastic science that's occurred in the past 10, 15 years. We're just bringing it to market, and we should have a test that's specifically catered to women with dense breasts early next year.
Incredible. And so you mentioned something quite fascinating, and this was a misnomer that I believed, that when I was diagnosed with early stage breast cancer, that I would progress through all of the stages before it metastasized, right? And as I am doing this important work, realizing that women are being diagnosed even metastatic right away, de novo metastatic, right? And so you're not necessarily going from stage 0, 1, 2, 3, and 4 and kind of these building blocks. And so what you're saying is that early on in these processes, like rogue cancer cells are kind of leaving their natural environment and going to other parts of the body, um, potentially undetected, but through the tools that you're developing and the science that you're researching, that there may be ways to find this and catch it very early.
That's exactly right. And this was some discovery that initially was in Germany and then has been replicated across the globe, where they found, incidentally, in a woman with DCIS, so this is stage 0 as we understand it, that cancer cells were escaping. And they found it by accident. They had done a bone biopsy in the femur and noticed when they did some genotyping of these cancer cells, they were anticipating they might see some leukemic signals, but instead what they saw were breast cancer signals. And indeed, when they performed a scan, they did find evidence of DCIS. And since then, that topic has led to a lot of research. There are some fantastic teams in the country that are involved in understanding the mechanisms. We published some work on this last year, where we showed there is a hormonal component to the escape of these cells, not just in breast cancer, but also in prostate cancer. So there are some regulatory mechanisms that allow these cancer cells to escape. The challenge, of course, is there are, for example, if you took a tube of blood, there are about 10 billion cells. If you look at red blood cells, white blood cells, all sorts of different types of cells, there are about 10 billion cells, and you're looking for 1 or 2 cells. So it's a huge challenge in trying to find that needle in a haystack. And for us, that challenge presented an opportunity. It was worth pursuing, because if we could truly find these early cells, then we can catch disease when it's the most treatable. And that's what led to our work in the past few years.
That's incredible. So I have so many questions to dive into with this. It's from also with our podcast and our webinar series, really trying to provide education in understandable terms so we can empower the patient so they can advocate for themselves if and when this test becomes available. And then also for our medical friends to let them know that there's really novel science coming down and can really change the way in which we are screening for not just breast, but now you're mentioning other types of cancers as well. So to take us kind of step back for our listeners who may not know necessarily what dense tissue is, can you give us a definition of understanding what dense breast tissue is, why it's important to know what our risk is, and how that leads to an increased risk of breast cancer?
Absolutely. So density, mammographic density is fundamentally how the images appear in a mammogram, how the breasts appear on a mammogram. The work started in the 60s. There was a different classification, but it was some of pioneering work in the 60s that showed that how the breast tissue appears in a mammogram correlates to risk. The true BI-RADS definition of ABCD appeared in the 90s, and since then it's become global. So breast could be one of 4 categories: A, B, C, and D. C and D categories are dense tissue. A and B are not dense tissue. And density comes from thinking about the amount of fibroglandular tissue in the body. So if you think about breasts, they've got glands that produce milk, they've got ducts that carry that, we've got fatty tissue around it, and all sorts of other connective fibrous tissue. So the amount of glandular material alongside the fibrotic material gives us a sense for density. Greater the amount of fibrous material or glandular material, more dense the tissue. It's a perfectly normal condition. Half the women in the country will have dense breasts. So that's almost 42 million women above the age of 40, between the ages of, 40 and about 80. That's 42 million women. They all have dense breasts. Dense breasts are a challenge for 2 reasons. One, our standard tools for screening, mammography, make it harder to spot. So it's harder to find these early cancers in dense breasts. Second, density itself is a risk factor And off late, we're learning that density could be a higher risk factor than familial history as well.
Interesting.
So it could be one of these dominant risk factors that we have to watch out for. Until the kind of work that Astrin and other teams are doing, there weren't tools that would allow us to find cancers early effectively unless we went in for an MRI for everyone. And so this ability to find cancers early in women with dense breasts not only reduces their risk, but also improves our outcomes. So it's a double whammy. Some have described it as trying to find a snowball in a snowstorm.
Yep.
And others have described it as just trying to find a bunny in snow, all sorts of different things. It's very hard to spot if you've got a lot of different white dots in a mammogram.
Yes.
That's what density is.
And it's so interesting that we're now understanding the correlation between density and risk.
That's right.
Can you share any hypothesis or science around what it is about the density that leads to that higher risk? Is it hormonal related or why? I used to think, because I didn't actually understand the correlation initially. I used to think, oh, well, of course, if we can't see it, then that it automatically just increases your risk because it's going to be detected later. But there's actually, outside of just the screening modalities, there's actually a correlation.
That is exactly right. It appears to be there are several different factors. We do not have a clear picture as to why dense breast presents itself with higher risk. We know it presents with higher risk, almost 1.5, 2 times the risk that non-dense breasts present. But why does it present so? We're not sure. There are many leading hypotheses, several studies pointing to, for example, it's a polygenic inherited risk. So it's not just one gene, it's several different genes that contribute to density. So some of it is just passed from mother to daughter. Then you've got just the age of just hormonal development. We're starting to see some shifts out there between when breasts start to develop and when young girls and women have their first period. So that shift and greater exposure to hormones also contributes to higher density. There are other risk factors, for example, that lead to density being prolonged across the life of the individual as opposed to shifting post-menopause. So there are several different mechanisms. I don't know that we have today a clear picture as to this contributes to exactly why dense breasts have a propensity or higher risk of developing into cancer.
Interesting.
But that said, there are fantastic models. The Tyler-Cuzick model is one of those models. The BCSC model is another model where we've got to start thinking about breast cancer risk as an individual, from an individual's perspective and an individual's circumstances, as opposed to thinking about it at large as a population. Right. So an individual who might have a variety of different risk factors, whether it's familial, whether it's hormonal, whether it's density, all these different risk factors will contribute into that model. And those models are now very accurate in predicting lifetime risk and 5-year risk. So knowing what an individual's risk is as early as age 25, especially if there is this history in the family, I think helps a lot.
Amazing. Thank you. Thank you for that clarity and definition and helping us understand, honestly, like this cutting edge opportunity, because when I was diagnosed, we never thought it would be correlated to density. It was always, the family history, the genetic testing, and all of these, we still don't know why. And I know there's so many of us out there who are just looking for some answer. I think I've come to the reservation of they just haven't found the gene yet. It's something out there. It's environmental. It's something we just haven't discovered it yet. But it's, it can happen to anybody, and we're still researching and figuring it out.
We're not going to give up. I know our team is motivated by that question.
Exactly.
Is to find it early, but also is to understand the underlying mechanisms as to why is this presenting itself with a risk? And more importantly, what could we do? Screening is one, early screening is one, easy screening is one, accessible screening is one, low-cost screening is one. So certainly by improving screening and accessibility, we certainly can improve outcomes, but also if caught later, what vulnerabilities exist, what pathways are expressed such that we could say, we've got this drug that was built for something else, but that might be reprioritized. That would be the best case scenario, is that we would have effective drugs and effective treatment modalities that even if we missed catching it early on, we can tap into it. So there are other groups who are trying to solve the same problem, but our whole existence is to try and address those questions.
So you mentioned, we're talking about breast cancer, and I also know that you mentioned prostate as well. So, how does this expand beyond just breast? Are we going to be in a world where, through blood tests, we'll be able to detect all early cancers?
That's the goal. That's absolutely the goal. There are certain pathways that affect majority of the cancers that we know to exist. And the idea is this 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 phenomenon that cancer cells leave the primary early is not just restricted to breast or prostate. We're seeing evidence of that in other cancers as well. If these cells are starting to escape, similar to a comet leaving a trail back, it is crosstalk because the immune system is looking at this and saying, well, hold on, what's going on? I'm going to catch this invader. And the cancer cell, in trying to survive, is going back and saying, no, I'm not an invader. So could we tap into that conversation and pick up these signals? So
Yeah.
we believe it is certainly doable. It's going to take a lot of work to get us from point A to point B, but that's the hope. That's the dream.
Lovely. So, I have to ask, we talked a little bit about this being in currently at the time of this recording, like in the clinical trial phase, right? And so, tell me a little bit about where you are in this clinical trial, the samples that you've collected thus far, just through blood work. From my perspective, if I can just draw blood during a routine checkup with my doctor versus scheduling the mammogram and all of this other painful screenings that we go through, please sign me up. I would be very interested. So, tell me a little bit more about how your trial came to be, who has been participating, and if there are any preliminary findings that you're able to share at this phase.
So there are several trials that we have run, each increasingly larger in terms of sample cohorts. We want to make sure that we aren't biased towards a particular age group or a particular individual type or a particular subtype of cancer. So we keep running increasingly larger and larger trials. Our most recent trial is going to have about 1,500 women split between healthy and cancer. So it's going to be a fairly large cohort. The performance we're seeing is truly extraordinary. We're starting to see, for example, this might be one way of framing it. For every 1,000 women with average risk, there are approximately, MRI would find approximately 17 cancers, breast cancers.
Wow.
So for 1,000 women, MRI would find about 17 cancers. In that same cohort, 3D mammograms, especially if they're dense, would find about 4 or 5 cancers. So there's a big gap between what an MRI or contrast-enhanced mammo finds versus what could be found in mammography. 2D will find slightly less. 3D will typically find 1 or more cancers. Adding ultrasound might increase that by a couple cancers, but there's still a significant gap. The signals we're seeing are sensitive enough that we can find 15, 16 cancers out of the 70 that are present. It's almost 3 times as many as what we've seen in mammography. So it could be a very, very powerful tool.
Yes.
Trying to find these cancers early. And all it requires is a standard blood draw. So a small amount of blood typically will get shipped to our lab, and we would process it using technologies we've developed in the lab. There's a lot of computational work that happens in trying to discern signal from noise. So it's the type of AI that we've been building over years that allows us to find these very, very faint signals that are present in early cancers and sift them from all the noise and accurately identify that this individual might have something.
So if the blood work comes back showing, a positive sign for this cancer, what are the next steps?
Yeah. So the next step, given that MRIs are sensitive, this is where we could be smart in terms of utilizing healthcare resources appropriately and ensuring those who absolutely need access to an MRI can get it. So if our blood test is positive, that immediately puts someone in a very high risk category. We've found something suspicious, there's something lurking, let's go get an MRI next. So the partners we're working with on the imaging side, this would be a seamless process. If we find, unfortunately, something being suspicious, they would automatically get scheduled to a nearby MRI center, and then they would go locate the lesion, get the biopsy, and then proceed from there.
Okay, so there's direct follow-up.
That's exactly right. We don't want to lose time. We want that direct follow-up. Because the one challenge that MRIs have in general, at least in the studies that we've seen, is even though they're very sensitive, it almost doubles or triples the recall rate. So a lot more women will end up going through a process, a diagnostic journey that might end up with, well, there isn't anything there. It was suspicious but not lurking. Imagine if we were able to combine the best of both worlds. Extremely high sensitivity with extremely high specificity. So we have fewer recalls, fewer false positives, but are able to find the cancers. That's the goal. That's truly the goal of the kinds of tools we're building.
Yeah. Amazing. I love that. That's so exciting. And I'm looking forward to just not only how these trials are progressing, but having this come to market where it could really become, the new standard for screenings and supporting women's health.
For example, when I talk to my family members, it's that uncertainty. For example, Asian women are more predisposed to having dense breasts.
Okay.
And it's that uncertainty of, is something lurking? And the notification that the FDA has drafted clearly states that if the mammography identifies dense breasts, there could be something that mammograms have missed. That uncertainty, that anxiety could be just crippling and paralyzing. What do I do next? What should I be doing next? Because ultrasound, it's great. It'll find one or two cancers, but I could be missing something. Or if I choose to get an MRI, fantastic. If there's something going on, it might look, but MRIs might not be accessible for everyone. And we don't quite have the capacity in the country to serve, for example, 40 million women with dense breasts and get them an MRI across the board. So for us, when we were thinking about how do we design this test and what's the purpose it's going to serve, that word certitude was sort of ringing in our minds. Our goal is to provide that certitude, that certainty. So the test is called Certitude. It's about providing that certainty for women with dense breasts to say, we've done the best we can and we don't see anything here. And you can be confident based on all the data, based on the thousands of women and all the data that we've analyzed and the science behind this, that you can be rest assured there is nothing here. Let's come back 2 years later. Let's come back a year later and see if something's going on.
Yes. Do you see this playing out in practice where women would still get their mammograms, and then if they have been notified of their density, then they could be eligible for this blood test?
That's exactly right. Today, we'd look at this as supplemental screening. So we'd highly encourage women, because mammograms do work for women who do not have dense breasts. So for half the women who do not have dense breasts, it is quite sensitive. So we would still continue to advocate that get the mammogram, understand your risk, right? So whether it's for family members of survivors, whether it's for anyone young, the idea is by the time you're 25, understand your risk. What might be the risk factors? And are you in the average risk, low risk category, or are you at a high risk category? If it's at a high risk category, then let's get on. Let's make sure you're screened appropriately and more carefully from an early age so we can find these cancers. There's some troubling data that just got published last week that shows that very young women are starting to develop. There is an increasing incidence of young women in their 20s and 30s developing breast cancer.
Yes.
And other cancers as well, colon and pancreas, but breast cancer, the numbers are high. And again, there are several hypotheses that are related to hormonal changes, lifestyle changes, changes in when women are giving birth to their first child, all sorts of different factors. Not a lot we can change about that, but we certainly can have a more personalized screening program. We can identify the risk, and if someone's at a high risk, let's make sure we give them the best science possible. Again, mammography was developed in the 60s, and since then, there's certainly been an improvement in the technology, but it hasn't fundamentally shifted. So today's tools, whether we use radiographic AI and other mechanisms to try and identify, there have been some studies in Sweden and other places that have shown it reduces, for example, physician workflow time. It speeds up review. In some cases, it will find 3 to 5% more cancers. All of this is valuable. Finding any number of cancers is valuable. But what we're truly looking for is that step function change. We've got to go from finding 4 or 5 cancers amongst 17. How do we find 15, 16, 17 cancers? That's a 300% change. And that's what we're looking for. And that's where fundamentally looking at different technologies, understanding from a first principles perspective, what is the biology of the disease? And could we tap into this disease? And could we tap into these conversations? And there's, of course, going to be a lot of trial and error and failures that guide that path. For us, there were several moments when we truly felt we've hit a wall, but a lot to the team to keep pursuing and keep pushing and finding answers.
Yeah, it sounds like you guys are asking really the right questions and really taking challenges and flipping that on its head to be opportunities. How can we solve this problem? Which I really think is where we're at. And with AI, even in my own work and practice, I'm finding this is really just like the cutting edge and the tipping point of the acceleration of discovery and opportunities to really blend human-centered approaches with technology and the advances in science. And so kind of bringing everything together in this day and age.
Exactly right. We couldn't be doing what we're doing today 5 years ago. That's the pace of discovery. Again, as you very beautifully put it, we've sort of at these inflection points. Technology is starting to truly get to a point where we can look at some very sophisticated models in a short order. At the same time, the kind of diagnostic precision we have today didn't exist a few years ago. So by combining different technologies, whether it's robotics, whether it's optics, whether it's AI, whether it's some very clever biochemistry, by trying to bring these different things together in a multidisciplinary effort, I think we're able to unlock signals that just didn't exist. Even as of last year, didn't exist.
No, exactly. Every year we go to the San Antonio Breast Cancer
Right.
Symposium or the ASCO conferences, and it's just they can't come fast enough. Like a year, like everything is changing, especially with the studies and everything that's coming out and these peer review papers and everything. So it's really great work
Yeah,
that
we're
we're doing.
excited. I think we've submitted some data to San Antonio. We're hoping to hear from them soon, but we should be presenting some of our largest study data
Oh,
at some point.
fantastic!
Yeah, we're excited. It pans out. Yeah.
Yeah. So for someone who's listening right now, and they're like, "Oh my gosh, there's this opportunity. There's a blood test I can follow up on."
Yeah.
What should the patient do at this moment? They're listening to the podcast. They know they have dense tissue. Who should they talk to? Who should they reach out to?
So we're going to be very careful about this, just given the magnitude of the responsibility we hold.
Hmm.
We want to make sure that we roll this out correctly, make sure that there aren't any gaps in the follow-ups. So we're assembling our partners. We're going to go initially, at least in 2026, early part of 2026, the first quarter, we're going to go certain markets. So we're going to pick 3 to 5 markets across the country where we know we have established partners, but we know we can make sure that if anything's positive, then we can get them to imaging ASAP. And that
Hmm.
process is seamless. So
Great.
we're working with the right groups to make that happen. Our website is certitudebloodtest.com.
Okay.
That's where, and there's a sign up on that website. So if folks are interested in learning more or participating when we start announcing some of these markets, then we'd love to hear from them and make sure that they get scheduled appropriately.
Awesome. Great. And I'll put all of those links in the show notes as well so people can find the website and sign up and follow up accordingly.
Thank you.
Is there anything that we haven't touched upon that you would like to share? I feel like I'm almost at a point too where I don't even know what questions I should be asking to empower our listeners and patients and physicians as we're kind of crossing this next phase of diagnostics.
In many ways, it feels we're at the beginning of something, not at the
Mhm.
end of something. It almost feels like, for some of us, that we're now starting to unlock information that just didn't exist.
Mhm.
And with more information and smarter models and smarter drugs that are starting to get developed, I'm hoping that we'll get to a point where it's not as scary as it is today. I'm hoping it gets to a point where that fear of hearing that there could be some cancer just disappears because we'll have the tools to not only spot it early and intervene when the outcomes are fantastic, but also know that, okay, it's of a particular fingerprint. Here's the molecular fingerprint of this particular individual. Here's how we're going to tackle it. I don't think we're very far off. Again, it didn't happen overnight. I cannot take all the credit for this. In fact, we take very little credit for any of the work we're doing. It's because of years and decades of advocacy, years and decades of science in a lot of different fields that have come together to bring us to this point. Our team came together at the peak of the pandemic. And if you guys could remember, I mean, that was, you had to be in close quarters and you had to be masked and we were looking for what sort of tests are available and all sorts of different things. It really did take a very dedicated group of people that I'm grateful to work with every day to come together to say, we're going to set aside everything and we're going to focus on this problem. Groups like these, I think, will make a difference and will get us to a point where it's not going to hopefully be as scary as it is today. And not just for breast cancer, but for a variety of different cancers.
Well stated, very beautifully done, and gives us so much hope, which I think we always need when we're facing the challenges of either personal diagnoses or of family and loved ones, and even for our children and our family members and our friends, understanding our risk. So, JP, this has been a fantastic conversation. I really appreciate you giving us a crash course and not just the exciting and excellent work that you're doing, but in breast density, understanding how the progression of these cancerous cells can flee the initial site and go to other parts of the body. And this really exciting technology that's coming down the line for all of us. So thank you for your time today.
Thank you. Thank you for the opportunity.
And thank you, everyone, for listening to our show. I would like to acknowledge that all of the information on our podcast is from personal experiences and is not a substitute for professional medical advice. You should always contact your medical care team. If you are looking for specific topics or would like to be a guest on our show, please feel free to reach out. My email is laura@survivingbreastcancer.org. Our show is made possible by our nonprofit, survivingbreastcancer.org, and by our incredible donors like yourself. You can hop on over to survivingbreastcancer.org to sign up for our mailing list, make a donation, and explore the 400+ programs we offer each year at survivingbreastcancer.org/events. Until next time, keep on thriving.